Good morning, you guys, and good afternoon to seventh hour. I want to talk a little bit about the cell cycle. So by definition, the cell cycle is a continuous sequence of events that occur in the life of a dividing cell. Okay, let me show you a picture right here. This kind of gives you a good idea of what I'm talking about for the cell cycle. The cell cycle has three major stages. You've got your interphase right here. You've got interphase, you have mitosis, and you have what we call cytokinesis. So we're going to look at each of those. We're going to draw a few stages, and the sub should have passed out to you guys kind of a little packet that you can draw what happens in the cell cycle as I'm drawing. Please be aware that if I go too fast, this is on Google Classroom, that you can always A, watch the video again, B, look at the PowerPoint notes. Okay, so start off, why do cells divide? And we talked about this before. The volume gets bigger than the surface area. So when the volume increases in size faster than the surface area, the cell has to divide. The cell has to be able to get nutrients from one side of the cell to the other so the cell can survive. So the reason cells divide, that's one of them. Of course, they get too big. Some other reasons is just pure growth. So when you've got a kid growing into an adult, cells have to divide to get taller and to get bigger. Some cells have to divide to reproduce, and those are bacteria cells. Some cells have to divide in order to repair, so replace any lost cells due to injury or disease. So when you fall down and you cut yourself and you're all scabby, after a week or two, then that scab goes away, that dried blood goes away, your cells divide and heal your cut. Unfortunately, cancer is a result of cells that divide and divide and divide and don't stop dividing. And we'll talk about cancer a little here down the road. Anyway, some different types of cells divide at different rates. So skin cells every 2 weeks, that kind of makes sense. Red blood cells every 4 months. Liver cells 300 to 500 days. And the intestine, the lining of our intestine, those cells divide every 4 to 5 days. So depending on what the cell's job is and what it does, a lot of times dictates how often they divide. Okay, so let's get back to the cell cycle. We've got interphase, we have mitosis and cytokinesis. How long is one cell cycle? Well, it all depends on what sort of cell we're talking about. Skin cells have a cell cycle of just 24 hours. Bacteria, 2 hours. And our three stages, interphase, mitosis, and cytokinesis. Okay, so before we start talking about these phases, I need to talk a little bit about chromosomes and DNA, okay, because we're going to be talking about that. So I'm going to start with this picture. So first of all, in our cells, we have our DNA molecule. This DNA molecule is wrapped around these proteins called histone proteins. Okay, those histone proteins, the way they kind of align themselves, and the DNA is actually really kind of stringy. We call it chromatin. And then you've got this chromatin circling and like kind of coiling and recoiling, I guess I should say. And eventually these chromatin fibers get closer and closer and closer together, and we get what we call a chromosome. Okay, so when we talk about mitosis, we're going to talk about chromosomes and when a chromosome duplicates. This is an example of a duplicated chromosome. Okay, a single chromosome has a top part like this, kind of the center part, and then kind of a long leg. And this is where the DNA is. The DNA is all coiled up really tightly and condensed very tightly to get this kind of chromosome structure. At one point, before the cell hits like mitosis, the chromatin, or I should say the DNA is, we call it chromatin. It looks more like this, okay? Real long, thin, and stringy. And then eventually it'll coil up, and then we'll get these chromosome structures that look familiar to you guys. Okay, so when it comes to chromosomes and cells, first of all, a chromosome consists of two chromatids connected by a centromere. Okay, so this guy right here, this is a chromatid, and this is a chromatid, and the center portion that attaches to both of them, that is a centromere. Okay, so this is a chromosome or chromatid. This is a chromatid, and this whole thing is a chromosome. Okay. We have homologous pairs of chromosomes. We get one from our dad, we get one from our mom. They kind of match up depending on what genes are found in each chromosome. When you have a chromosome replicate or duplicate, okay, so this is one single chromosome. And when it duplicates, it makes this double, so it looks like kind of a fork of some sort, okay? So this side is identical to this side. So when a chromosome has duplicated, it's drawn like this. This is a duplicated chromosome. This side is identical to this side. And this is a sister chromatid to this. Sister as in they're identical. Okay, so chromatid is a half of a chromosome right here. DNA is going to copy itself to make a sister chromatid. So here's a chromatid, and here's the sister chromatid. Body cells are called somatic cells, and we have a total of 46, okay, chromosomes, or we could say 23 pairs. However you want to talk. If we're talking about single chromosomes, we got 46 or 23 pairs. Of course, one comes from dad, one comes from mom. In our sex cells, so in the sperm, there's only 23 chromosomes, single chromosomes, and then in the egg, there's only 23. So if you are getting 23 from mom and 23 from dad, you're going to end up with 46 chromosomes total in all of your body cells. Somatic cells are body cells. We always represent somatic cells with a kind of symbol. It's 2N. So if we're talking about a cell that is 2N, that means it's a diploid cell. Okay, diploid cell 2N. That means that it has the full number of chromosomes. So in humans, the full number of chromosomes is 46. Haploid, hap is like half. So our gametes, which are sex cells, are just N. So that's half of our diploid cells. We're talking about chromosomes. So all of our body cells have a total of 46 chromosomes or 23 pairs. Our sex cells only have half the number. We call them haploid. It was represented by N. Each of our sex cells only have 23. So when the sperm fuses to the egg, you get a total of 46, which is what we need in humans. Okay, so let's, now that you kind of have an understanding of chromosomes, let's take a look at the actual cell cycle. Okay, so there are 3 stages to the cell cycle, and I think... Okay, so let's go ahead and take a look at interphase. So interphase, as you can see from this diagram, is the longest stage of the cell cycle. There are 3 stages. There's G1, there's S for synthesis, and G2. G stands for gap or growth. Your book calls it gap. Other places will call it growth. So whatever you prefer is totally up to you. Okay, so there are three phases in interphase. There is growth 1 or gap 1, there is synthesis, and there's growth 2. So in growth 1, the cell is growing, it's developing, grow, proteins are being made, and organelles are making a duplication of themselves. The idea is this, is when mitosis occurs, and mitosis is basically cell division, okay, for body cells or somatic cells. We are going to start with one single body cell, and we will end up with 2 identical body cells. So in growth 1, okay, this is interphase. This is kind of the resting stage of the cell's life. So proteins are being made, cells grow, organelles duplicate. Synthesis comes along. Synthesis is not quite as long a phase as growth 1. The chromosomes are going to duplicate, forming 2 identical strands of DNA, or 2 sister chromatids that are held together by the centromere. So just a quick reminder, you've got your centromere, this is a single chromosome. So when it duplicates, it is going to look like this. Okay. At this point in synthesis, the chromosomes are really like, it's chromatid. Or let me start over. The chromosomes are not condensed like this into their little packages. The DNA is in the form of chromatin. So the DNA during synthesis is looking kind of like this, or actually more like this. Okay. So we call it chromatin before it condenses and it looks like a big old chromosome. So during synthesis, it's looking kind of more, actually probably more like this. Okay. And then as it starts to condense in prophase, which is mitosis, it's going to look like this, and then eventually it'll be packaged up and it's going to look like this. So right now in synthesis, in your head, you'll have to imagine the chromosomes looking like this. So growth 2, or gap 2, the chromosomes or the cells are growing, and then the centrioles make a copy of themselves. If you remember centrioles, we said they play a role in cell division. So now you're going to see what that role is. Okay, so you have interphase, and then you have mitosis. So mitosis is cell division of somatic cells. And we always start with one parent cell, and it divides into 2 identical, what we call daughter cells. Okay? And they are literally identical. Okay? If this was bacteria, this is basically reproduction for them. For humans and other animals, mitosis is cell division. When the cell gets too big, when that volume to surface area ratio is too large, the cell has to divide in order for it to live. So mitosis has got 4 stages, or phases, I should say. And then after mitosis, we have cytokinesis. So our 4 phases of mitosis are prophase, metaphase, anaphase, and telophase. So I'm going to switch my view, and I am going to move, and we're going to do a little bit of drawing. Okay. There we go. Okay. So again, you can have, or these notes are on Google Classroom, so you can take a look at them as if I'm going too fast. Okay, so I'm going to get a few different colors of pens to make things a little bit easier. So first phase in mitosis is called prophase. The chromatin coils into visible chromosomes, kind of that fork-like structure. Nuclear membrane and nucleolus start to disappear, and spindle fibers start to form. Okay, so I'm going to add one more. I'm going to say centrioles move to opposite poles. Okay, so I'm going to draw this. So this is how I'm going to draw it. I am going to draw my nuclear membrane. So it is a dotted line. So it looks like it is, that's a little bit better. So it looks like it's disappearing. Okay. I'm going to draw two sets of chromosomes. I'm going to draw this guy, and I am also going to draw one more. Well, that's not helping now, is it? That's not helping either. Okay, that's a little bit better. Kind of. Okay, so I'm going to draw two chromosomes. So by now the chromatin has coiled up, and so my chromosomes are condensed, and it's got this kind of butterfly looking, structure to it. My centrioles move to opposite poles. So I'm going to draw my centrioles like this. Okay. So my centrioles have moved to opposite poles. And these little microtubules called spindle fibers start to appear in the cytoplasm. They actually kind of come out of the centrioles. So major things that are happening right now, nuclear envelope starts to break down and it starts to disappear. The nucleolus disappears. The centrioles move to opposite poles, like this, and the spindle fibers start to appear from the centrioles. Again, they're like microtubules. And then our next phase, so there's not too much going on there. So our next phase is metaphase. So in metaphase, our nuclear envelope has completely disappeared, and our chromosomes are going to line up at the equator, or the center of the cell. So I'm going to start by drawing my centrioles on opposite sides. Okay. And now I'm going to draw my chromosomes. My duplicated chromosomes, okay, are going to line up at the center of my cell. Now, if this was our body cells, then we are going to have a total of 46 of them lining up at the equator. Okay, so our spindle fibers actually attach to our centromeres. Okay, and that is metaphase. Okay, not much going on. Oh, I'll put my a few more spindle fibers. Super easy to remember. Chromosomes line up at the equator, middle of the cell. Spindle fibers attached to the centromeres. So our next phase is anaphase. So the centromeres split apart. There's a separation of the sister chromatids, and the chromatids move to opposite poles. So I am going to draw my centrioles again. Okay. I'm going to draw my chromosome. I'm going to draw my spindle fibers like this. They start to contract. So I'm going to draw one chromatid here and the matching sister chromatid here. And I'll do the same thing with my red chromosome. So what happens is those spindle fibers start to contract, and those chromatids start to pull apart and move to opposite ends of the cell. As you can see right here, this is a real picture of a cell in anaphase. Those chromosomes are making their way to the opposite sides of the cell. Okay, so our last phase is in mitosis is telophase. So chromatids reach opposite poles, chromosomes unwind, spindle fibers start to break down, nucleolus reappears, and the new nuclear membrane starts to form. Okay, so our chromatids, I'm going to make our chromatids 1. Oh, I'm going to start over. Try that again. Let's make our 2 nuclei. Nuclear envelope starts to reappear. Our chromosomes, our chromatids have reached opposite ends. The spindle fibers start to break down. I'm going to put my centrioles back over here. My spindle fibers I am going to just make really light. Okay. Chromatids reach opposite poles. Chromosomes start to unwind. So I'm actually going to make my chromosomes a little kind of wiry looking, not so thick. So they're starting to unwind. Okay, that is, let's make our nucleolus. Oh, that's okay. That'll work. Okay, so our last stage is cytokinesis. That's really hard to see right there. Okay, let's look at these and then we'll move to the drawing. So cytokinesis means cell cutting. And this is where the 2 new cells with identical information are truly formed. In animal cells, there's something called a cleavage furrow that forms in that cell membrane and eventually pinches off to give us 2 new cells. If it's a plant cell, what happens is what we call a cell plate forms in the middle and separates the 2. And that cell plate is made a lot of cellulose because a cell wall has to be formed. So in cytokinesis, a cleavage furrow looks something like this. The cell kind of starts to fold in on itself. And I'm going to get rid of this so it looks like that. So this kind of folding in is called the cleavage furrow. And I'm going to put a dotted line because this is where our cytoplasm starts to actually divide. We've got our 2 nuclei over here. Our spindle fibers have completely gone away. I'm going to put my real thin chromosomes over here, like that. And eventually what happens is in animal cells, this cleavage furrow, these 2 come right together and it's going to form these 2 cells, just like these 2 cells. Okay. If it is a plant cell, what's going to happen is this. The cleavage furrow is not going to happen. Okay. Not a cleavage furrow. But right in the center, we have what we call a cell plate that forms. And that cell plate is part of the new cell wall. And we're going to end up with 2 brand new cells. So like our centrioles in an animal cell are going to move back to kind of the middle of the cell. You have a fully formed nucleus. You have your chromosomes that are very thin and not condensed any longer. Okay, my friends, like I said, this information is on Google Classroom, so if I went too fast, then make sure the sub stops the video and you guys can have some time to write more information down. Okay, hope all is well. You'll have lots of practice with this, so you'll be pros of mitosis before you know it. Thanks, you guys. Miss you guys. Bye.